this is a very simple architecture of the modeling part of EBRAINS, and I don't want to go into many details of the organization, but this is just to point out. Yes, you have a mouse here. To point out that we have a service for everybody, because I understand that when you are confronted with an infrastructure, you want to have things in general as simple as possible. So I understand that we are all scientists here, but we are some of you are experimentalists, some of you are modelers, some of you are what we call power users, and so there is a wide variety of expertise and backgrounds which we need to take into account. I've been teaching to both students in the neurobiology courses and the computer science course. And of course, I have different kind of audience. So when I'm talking with the neurobiologist, usually when they see an equation or when they see a computer, they begin to shiver, okay? On the other side, when I'm doing with computer scientists, they have no idea what ion channel is. So we are trying to take into account all this expertise in running something without downloading anything. This is the key point. When you are using EBRAINS for simulation, you don't download on your computer, you don't install or pip install any software on your computer. You go through the platform, through a web browser, and do everything. And so we have, so we have three types of different users: learner, beginner, a normal user, and expert. And according to their expertise, they go to different kind of use cases. There are use-specific, very simple use cases in which you just point and click. And I don't know if I have the time to show you some of those. Others are requiring some kind of expertise because they need to interact with the knowledge graph. Maybe you have a Jupyter notebook, so you need some kind of coding expertise in Python. And there are also some other more specific use cases in which you basically what you need is only a computer resource, either because you have a praise grant and so you have a large amount of computer time that you can use, or because you just need a small amount of computer time and you don't want to go through the process of asking for a specific account. You can do this also using the supercomputers that are attached to the Human Brain Project and the EBRAINS platform, which are one in Switzerland, the Lugano, the CSCS. In Bologna is the Cineca, the CEA in France, and of course ULIC supercomputer systems. All of those are accessible, and for one of those, in particular the CSCS system, we have also set up what we call a service account. A service account is something that allows you to use transparently computer resources and launch a parallel job without having an account, because we take care of monitoring and checking and giving you a limited amount of resources to run the supercomputing jobs. And okay, so you have seen already this kind of website. I hope you already have some experience in navigating it. I'm focusing on the services that are going to be under simulation, okay? And there are different kind of levels. Cellular level simulation, the one in which I'm most involved with. Network level, using essentially NEST. Whole brain level simulation, this is the virtual brain of Victor Girsa field. And data analysis and visualization. I'm going through the slides first, and then if I have time, I'm going to link to the internet to go on an interactive visualization of a few use cases. Okay, so these are the. So I have the kind of glasses which don't allow me to look at the screen here and I can't see there. So okay, so this is the list of type of simulation that you can do in the simulation part. And you go from neuromorphic computing to the neurobotic platform and other examples in which you can do different types of simulation levels, okay? In some cases, you have structures and applications that allow you to run something. In other cases, unfortunately so far, but this is changing rapidly, you need to download something still. But I'm going to focus a little bit on the interactive workflows for cellular level simulation, because we've been involved in building this part of the platform. But you can go on and check anything here. So what can you do on the brain simulation platform for the interactive workflow at cellular level? You have different things. Trace analysis. This is very simple. You don't need anything about this, because there is some coding involved, but this is already done by us. So you go there and do what the experimentalists do when they want to analyze traces, electrophysiological traces. So you want to capture, for example, the electrophysiological features of the traces and do the average, not only using your own data, but also catching data from the knowledge graph. And so you can put together the data that we have on our databases with the data that you have just recorded. And you can make comparison, extract features. There are about 200 kind of features that are available for being analyzed. AP, duration, amplitude, spike times, and interspike intervals, voltage levels, and everything. Single cell modeling will allow you to build your own model of single cell in a realistic way, which means having dendrites, morphologies, ion channels, distribution, and everything. Morphology analysis, single cell in silico experiments in which you have a network, or no, in this case, a single cell, and you can put an electrode and do an in silico experiment at the single cell level or at the circuit level. You have a circuit. In this case, I think that so far we have hippocampus, of course, because I'm working on it, and also cerebellum from the GDO D'Angelo Labs, and the cortex from Henry Markram. So we have a few networks, realistic networks online, and you can run in silico experiment. When I say in silico experiment, I mean multi-site recordings, stimulation, and synaptic interactions and things like that. And also you can run networks, realistic networks. We have, for example, the rat CA1 region in full scale. It is 500,000 neurons with the dendrites, axons, and it is available to run simulation there on the current version of the model. This is an example. These are some views from the use cases. Now, if I have time, I will go through it interactively. So this is a list. And you see here a very important icons. Those icons will allow you to understand before you run the use case. They would allow you to understand what kind of expertise you need to run those experiments. Okay? And this is important because if you are a beginner or an experimentalist, never done a modeling so far, it is kind of difficult to choose one of these use cases and then don't know what you do, because of course you need to do what you are doing. We don't check what you're doing. So it may be difficult at some cases, but in many cases you will see also that there is an online interactive video. It is a video which is guiding you step by step through the entire use cases, allowing you to make choices and say, okay, I want to do this, then you go there, you want to do this, and you go there. It is a very simple explanation of what you do step by step. In this case, this is the in silico experiment. This is the small circuit in silico experiment. We have come up here with the entire model of the CA1 hippocampus in the rat. You can select neurons where you can put an electrode, a recording electrode or a stimulating electrode. You can set up the specific synaptic activation, and you can run an in silico experiment, or you can configure and run a simulation of the entire system, on a supercomputer, and then you get the results. You can analyze the results. You can get different types of analysis. This is another resource that we have put online. This is still not in the EBRAINS website. It is online on its own, but it is going to be part of the EBRAINS. It is what we call Facility Hub. There is another Facility Hub for the cerebellum, and here what you will be able to do, and I hope that I will have time to show you something, is to cherry picking elements of your model to put together a single cell model. So for example, you go from the website, you can go and choose a morphology from the neuromorpho.org. I don't know if you know this website, but it is a very popular website where there is a list of morphologies, 3D morphologies of different neurons in the brain. You can pick a morphology from neuromorpho. You can pick the ion channels from ModelDB. You can pick the data from the knowledge graph. You can put everything together and arrange automatically a workflow to do the model of the single cell that you have chosen. Okay, so we are inside the workflow use cases of the cellular level simulation. So just to go from the simple stuff to the more complex stuff, let's go to trace analysis. Trace analysis is here. I hope that it does not ask me for something. Yeah, okay, so let's see. So in this case, for example, we have a set of data that are coming from the knowledge graph of different laboratories, so we can choose whatever we want for analysis. So for example, I'm going to use Alex Thompson. Rat is okay, hippocampus, CA1, interneurons, pyramidal cells, let's say pyramidal cells, and then this type of E-type, and this should come up with the traces that we want to analyze. Of course, as I told you, you can also download your data, and these are the traces, and these are all the traces that we have. Suppose that we want to select a few of them, and then you can go here, you can select all, or just a single trace here and there. Okay. Just select all, it's okay. And then you can go on and select, and then add the cell, no, go next. Yeah, I agree, or whatever you say. Confirm, yes, I confirm. And then you can select the type of features that you want to analyze, okay? So I will not go on with this because I want to show you something else. So let's go back to other use cases. Okay, I can close this. You see here, for example, you see here that the icon says beta version, which means that this model has not been released for the public yet because there is not a publication with it, so we may have the right to change it in the future. Everybody, because this is a point-and-click interface, and this is a web application. So basically anybody can run this without even having an account, okay? Because this is run on the internal services. I hope so. Let me see. Otherwise, yeah, it is coming out. Good. Here we go. And of course, this is the graphics. Nah. I really need a Windows computer here. But if you can go online whenever you want, and you can check it while you are doing this, and if you click on the mouse, you can rotate and look at different things. These are all the cells. I can. I don't get access here to this Windows, but you can do the simulation here. I cannot do it. Well, let's try something strange. Okay, simulation configuration. And I need to add the recording. No. No, I cannot do it from here. Anyway, never mind. Trust me, it works.
But only Windows.
No. Also in Mac, it's a web application, but I need from this computer. I don't have the access to the browser. I don't know why. And I think that I'm going to stop here and maybe asking questions if there are. Okay, thank you very much.